Hello, this is Mr. Stubbe again, and this is our second screencast from chapter 19.1. We're basically going to talk about the second part of section 19.1 today. We talked last time about Carl Linnaeus and his system of classification. He thought it was very important that not only are all species named, but also they would be put in 7 different categories. And we're going to start off here today. Can you recall the 7 classification groups? We use a little memory aid called Kids Prefer Candy Over Fresh Green Salad. And see if you remember what those are. Okay. And here they are again. It's Kingdom was the largest group, Phylum, a little smaller, Class, Order, Family, Genus, and the smallest group would be the Species. So again, these are groups within groups. Several species belong to a common genus. Several common genuses or genera belong to a family, and so on, all the way up to the kingdom. Over time, we realized that this classification system would be changed a little bit, beginning with the number of kingdoms that we had in the first place. So let's take a look at some of the ways in which we've changed our thinking. Back in the 1700s, when Linnaeus first designed a system, he had only 2 kingdoms. We called them the plant and the animal kingdom. Basically, if you sat in one place and you seemed to grow on your own, you were a plant. If you moved around, you were an animal. By the late 1800s, of course, with the invention of the microscopes, we realized that there were all these little microscopic things as well as the plants and animals. We created another kingdom called the Protista. As microscopes got better, we realized that there were more than just one type of single-celled organism. We had the bacteria, which were in the group Monera, and then the others, larger ones, which were eukaryotes in the Protista. These would be things like the algae and amoebas and so on. Also, over in the plant area, we realized that there were these mushrooms and other kinds of fungi that didn't really do photosynthesis like the rest of the plants, so we separated them out into their own kingdom as well. By the time the 1990s roll around with DNA evidence and other techniques, we realized that this Monera group or bacteria group could be further subdivided as well into the eubacteria, which are basically most of the bacteria we know of today, or that I should say that are common today. And then there's these strange ones called archaebacteria, which basically look the same as eubacteria, but they're very, very different in terms of their DNA. We kept the other 4 kingdoms. So by the time this is rolling around, some people thought that this was just getting to be too many kingdoms. So what we ended up doing was creating a new category called the domain. So this is a bigger, broader group even than the kingdom, and this was developed in the 1990s. So today we have 3 domains, okay? The one, bacteria, which is also a kingdom. We call it eubacteria kingdom. Archaea is another domain. We call this Archaebacteria kingdom. These are both the same as their kingdoms, the same groups. And both of these are prokaryotes, meaning they don't have a nucleus. And then everything else, the plants, animals, fungi, other protists, and so on, are grouped in this domain called eukarya, and they are all the eukaryotes. So these are our 6 kingdoms that we have today. Again, this eubacteria is also a domain called bacteria, and then this archaebacteria is also a domain called archaea. These other 4 are grouped into the domain eukarya. They have a nucleus. So what I'd like you to do at this time in your notebook, hopefully you have that out at this time, is for each of these kingdoms, we're going to have you describe the following characteristics. I would maybe make a chart in my notes if I were you. You can use the textbook website in section 19.1, the e-text. What I want you to do is for each of the kingdoms, describe the cell type, basically whether it's a prokaryote or eukaryote, how many cells they have in a typical organism, multicellular or unicellular, the major cell structures, method of nutrition, how they eat, and then 2 to 3 examples. I said from around the room. This would be assuming we were in the classroom, which we aren't. So maybe you have some around your home or others that you might know of. So you can pause the video at this point. I'm going to continue on here in a second with characteristics of these kingdoms. with me for a moment here. I'm pulling up another slideshow that I have. There's a little joke for you. What do you call a fish with no eyes? A fish. All right, here we go. No, this is the wrong one. There's that joke again. Okay. So we'll start with Kingdom 1, and they're in no particular order here, but the eubacteria, oops. Okay, pointer in here. Kingdom 1, the eubacteria. These are all autotrophs or heterotrophs. In other words, they can get their food through photosynthesis or by eating. They are all unicellular. Even though in the picture here it looks like they're multicellular, these are actually unicellular ones chained together, sort of forming colonies. They're all prokaryotic, meaning they don't have a nucleus. Here's a real important one. They all have a cell wall with a chemical in them called peptidoglycan. This was kind of an early discovery and something that gave them an early clue that this bacteria kingdom was really different than the one we're going to talk about next here. Okay, this was a chemical in their cell wall. Some of them are mobile, meaning they can swim around, and these are what we call the true bacteria. E. coli is one example. These are bacteria in our mouth, on our hair, on our surfaces. So this is what most people are talking about. We're going to spend a whole unit on this in a couple of weeks from now, so more on them later. Kingdom 2 is the archaebacteria. Again, originally they looked very similar under the microscope, but DNA evidence showed that they were very, very different. They too have autotrophs and heterotrophs. They're unicellular. They're prokaryotic, but here's the big thing. They do not have peptidoglycan in their cell walls. Okay. Some of them are mobile. These are very ancient bacteria. They live in really extreme environments like this ocean vent. Some of them are methanogens, which basically means they generate methane gas. Halophile. Halite is salt. Phile means to love, so they live in real salty places. Thermoacidophiles live in very hot and acidic places, so real extreme environments, such as this hot spring that I took a picture of in Yellowstone National Park a few years ago. Kingdom 3 is the Protista. Not a very good kingdom right now because there's so many different things that are in it that just don't seem to fit anywhere else. There's animal-like things in there, fungus-like, plant-like. This is a kingdom that biologists are really trying to reorganize right now. I sometimes call it the junk drawer kingdom. It's things that don't really fit anywhere else. So here's some of this, like red algae, for example. It's an autotroph, but it's red. It's not typical green. Here's a single-celled organism called an amoeba. Here's an animal called a euglena that actually swims around like an animal, but it also does photosynthesis like a plant. So where do you put it, plant or animal? Well, they don't know yet, so they put it in the Protista kingdom. This other kingdom is fairly well organized. It's called Kingdom Fungi. It includes some very important decomposers like this mushroom here. All of them are eukaryotic. They can be unicellular like this yeast or multicellular like this mushroom. But here's something to keep in mind. All of them have a cell wall made of a chemical called chitin, C-H-I-T-I-N here. Unlike the peptidoglycan, which is in the bacteria, this is a different chemical. So even though the cell wall looks similar under the microscope, it's very, very different in origin. Some fungi can be helpful and delicious, such as mushrooms. Some are actually beneficial for creating drugs, like penicillin actually is made by a fungus. Okay. Some of them cause diseases, some of them just irritating, some of them very harmful. This one is the athlete's foot disease. Here's their little cells called hyphae, which grow into your skin and make you itch. So keep your feet dry. Ringworm is very similar. Here's those hyphae that can grow into the skin and create little rashes like this, kind of irritating when they get into those cells and irritate nerve endings. Fungi are all heterotrophs, meaning they cannot make their own food. Most of them are decomposers. They get their nutrients from dead organic matter, dead wood, dead animals. Some of them are parasites, like that athlete's foot we just mentioned a moment ago. And this next kingdom is my favorite one, the plant kingdom, or Plantae is its Latin name. Most of them are multicellular. They have another kind of cell wall now, the third kind of chemical we're going to talk about, called cellulose. Okay, so it puts it in its own kingdom. They're autotrophs. Whoop, skipped right to my animals here. Last one is the animals, as you might have guessed from the pictures. All animals have a couple things in common. They're multicellular, lacking a cell wall. Whoops, I forgot to delete that space there. They're all heterotrophs, and most are capable of some kind of movement. Since they can't make their own food, they have to go to it and get it. So this puma here is obviously stalking its prey, jellyfish moving to find prey, and so on. And this little grasshopper or katydid also. So we're going to conclude at this point, okay?